Assalamualaikum Dr. Amaliawati and Dr. Naziz. My name is Syazana Adilah Binti Zulhaidi, and today I will be presenting my study titled Plant and Microorganism-Derived Agent as Biofilm Inhibitors Against Predominant Oral Pathogens, a review. Before we dive into the details, let me introduce you to our study mentioned earlier. Did you know that the oral cavity hosts over 700 distinct microbial species? This makes it the second richest microbial environment in the human body. Among these species, Streptococcus mutans, Porphyromonas gingivalis, and Candida albicans are particularly notable, which typically considered commensal of the oral cavity. However, under certain circumstances, these microorganisms can become pathogenic and contribute to various oral diseases by forming biofilms. Biofilms are structured communities of microorganisms embedded within a cell-produced matrix. One of the challenges with biofilm is that they exhibit increased resistance to anti-biofilm agent compared to planktonic cells. This resistance makes dental treatment significantly more challenging. Therefore, this review highlights plants and microorganisms which demonstrated promising antimicrobial and antibiofilm activities against major oral pathogens in vitro. For example, cranberry proanthocyanidins inhibit biofilm formation of Streptococcus mutans by disrupting bacterial adhesion to tooth surfaces. The widespread use of antibiotics such as penicillin, cephalosporin, and metronidazole in dentistry has led to significant issues, including the rise of drug-resistant bacteria and the disruption of the oral microecology. Therefore, there is a crucial need to explore alternative substances that selectively target and dismantle oral biofilms, thus preventing oral diseases. The three main objectives of this study are to analyze the predominant oral pathogens that form biofilms in the human oral cavity and their roles in oral diseases, to determine the plant and microorganisms-derived biofilm inhibitory agents and their effectiveness in combating oral pathogen biofilms, and to review the mode of action of plant and microorganism-derived biofilm inhibitory agent. Under the first objective, there are three most prevalent oral pathogens. Firstly, Streptococcus mutans, which is a key contributor to dental caries because it produces extracellular glucopolymers from sucrose, which helps it colonize teeth surfaces. It is highly acidogenic, converting various carbohydrates into organic acid, lowering the pH, which contribute to dental caries. Additionally, as mutants secretes mutacins that inhibit the growth of competitive bacteria like Streptococcus sanguinis and Streptococcus gordoni, allowing it to dominate the oral biofilm. Secondly, Porphyromonas gingivalis, a major cause of periodontitis, primarily living in the subgingival sulcus of the oral cavity. It has various virulence factors such as fibrae, capsules, and gingipains that contribute to its pathogenicity. The bacterium causes periodontitis by producing toxins that inflame and damage gum tissue, leading to the destruction of the structures supporting teeth. Third is Candida albicans, which is the most common fungus in the oral mucosa and can cause oral candidiasis, especially in immunocompromised individuals. It forms biofilms by attaching to myelin receptors on its surface, often with Streptococcus mutans, leading to increased biofilm accumulation and tooth decay. The shift from yeast to hyphal form marks its transition to a pathogenic state, contributing to virulence and biofilm formation. Thus, plant-derived anti-biofilm agents, which include essential oils, flavonoids, and tannins, are effective against oral pathogens, offering benefits such as lower cost, minimal side effects, and reduced resistant development compared to synthetic counterparts. For instance, frankincense, extracted from Bowisia serrata, inhibit the biofilm formation by Porphyromonas gingivalis with minimum inhibitory concentration of 32 microgram per milliliter. Their mechanism will be discussed later in this video. Besides, microorganism-derived anti-biofilm agents also have been recognized as rich sources for novel antimicrobial agents due to their unique biological properties. Microbial anti-biofilm agents are highly specific against biofilm-forming microbes. They are also biodegradable and environmentally friendly. For example, leomycins of fungus Aureobasidium pullulans can inhibit biofilm formation by Streptococcus mutans. There are four major mechanisms of natural anti-biofilm agents emphasized in this review. First is the disruption of microbial cell membranes. Bacteriocins, and antimicrobial peptides from Lactobacillus plantarum form pores incorporating bacterial membranes, resulting in ion leakage and cell death. Plus, essential oils like oregano, thyme, and clove contain active compounds such as carvacrol, thymol, and eugenol, which increase bacterial membrane permeability and cause cell lysis. Second is the inhibition of quorum sensing. Quorum sensing facilitates microbial communication, and disrupting it is an effective strategy to prevent biofilm formation. Anti-biofilm compounds interfere with quorum sensing by either inhibiting signal molecules or imitating the signaling molecules to block their respective receptors. Thirdly, the degradation of extracellular polymeric substances (EPS). The EPS forms the structural matrix of biofilms, providing stability and protection to the microbial community within. Microbial enzymes such as amylase, cellulase, and dextranase can degrade the EPS matrix of biofilms. Plus, plant-derived phenolic compounds like curcumin can disrupt the biofilm matrix of Porphyromonas gingivalis. Lastly, inhibition of bacterial adhesion. Many natural compounds can interfere with the initial attachment of oral pathogens. For example, a microbial-derived agent, including biosurfactants like surfactant, was reported to promote the antimicrobial activity of terpinone for all (TP) against Streptococcus mutans, which can inhibit bacterial adhesion when combined with TP. In conclusion, the objective of this study have been achieved. We explored numerous agents from plants and microorganisms that can prevent biofilm formation by oral pathogens, aiming to reduce reliance on current antibiotics. These natural inhibitors can reduce oral pathogens' ability to form biofilm, decrease their virulence, and enhance their susceptibility to antimicrobial agents. Currently, we lack understanding of how plant-based antibiofilm treatments affect the host's natural oral microbiota. This can be addressed by utilizing in vitro models that mimic the oral environment to help study interspecies interactions. Moreover, there is a lack of clinical trials evaluating the real-world effectiveness of plant-based antibiofilm treatments in oral care. To address this, we need to develop and conduct extensive clinical trials with diverse participants and following standardized protocols to monitor clinical outcomes. And with that, I conclude my presentation. I would like to extend my gratitude to the panel members, Dr. Amaliawati and Dr. Naziz, for their attention. Thank you.